And then translation.
Great.
Thank you.
Great. So replication, there's a central dogma. We have it as replication. That's a copy of the DNA being made to, copying a segment of the DNA, that is the gene, to form the mRNA. And the mRNA, that is transcription. And the mRNA will be sent into the cytoplasm to, produce proteins. And that process is called translation. So replication, transcription, and translation. Okay. So if we are talking about proteins, then we have to talk about types of protein, or the forms of protein, the structures that we have. So we have proteins are the biomolecules made up of long chain of amino acids. We have looked at that already. Long chain of amino acids. They are essential molecules found in all living organisms, except that you don't eat certain living organisms, but all living organisms are edible, including ants, including termites, including weevils, including maggots. They are all organisms that are eatable because they all contain proteins through to all plants. But the caution here is that some of them produce certain chemicals that are not edible. So if you're able to pull out this, or better say, if you're able to take out these chemicals that they use for their, defending themselves in the habitat or in the ecosystem, then you are good to go. You can eat them. Snakes and all that are eatable because they contain proteins. So feel free and enjoy everything.
The Chinese are happy.
Prosper, are you
Come
listening?
again.
Take note. Oh.
Oh yeah,
Hello,
I'll take the
hello,
I'll
sir.
take the termite.
Hello,
So
sir.
Yes,
that is
boss.
why the Chinese eat everything.
The
Yes.
wing
Yes.
reproductives, they are sweet like something.
Yes,
Hello,
boss.
sir.
So I know some. If you don't eat
Hello,
it,
sir.
you don't have to condemn it. Yes, sir.
Akokono is one of the best maggot that you can get.
Yes, sir. Yes, sir. I've eaten some
Akokono is
before.
very, very, very proteinous. So
Yes,
tell our
I've
ladies to
eaten
learn how
some.
to prepare it.
Yes, sir.
So
I've eaten
oily.
some.
So oily.
I appreciate your concern, sir. All right. So let's look at proteins. The main structures of protein. So first, we are looking at primary proteins. We have also the secondary structure, and then we also have the tertiary structure proteins, and then we have the quaternary structure proteins. But here, I have some assignments for you. So let's look at some assignments. Unfortunately, the submission date, I would have to change it and move it a bit. I thought this would be taught last Saturday, so that's why I made it 25th October. But then, since we've exhausted some of the days, I'm going to move it to next week. Please, next week is which date? Can you give me the dates for next week, Saturday?
That will be 1st November.
Okay, that's fine. 1st November. That's fine. Thank you very much. So, this submission date will be 1st November. And the question is, discuss the levels of protein conformation and give specific example. Discuss the levels of protein conformation and give specific examples. So you send it to my institutional mail. So d.baskobna. Please check the email very well, because you will come back to me again and be complaining that when you send, you are not able to send and all that. Check. It's d dot bat dash kobina at stu dot ucc dot edu dot gh. So please, as you send, take note of the email, and when you are typing, you don't leave anything out, because when you leave anything out, it might not come. It might not come. So tell the other colleagues as well. If you leave anything, it might not come. So tell the other colleagues as well. Okay, so submission date, I'll change the date to 1st November. So submission date is 1st November. Kindly alert other colleagues on the page who were not able to join. And everybody must submit it. Everybody must submit it to my mail because I'll use it to grade you as well as your assignment. Okay. Thank you very much. All right. So let's look at functions of proteins. What can we use protein to do? The body. What does it use the protein to do? Then number one.
Hello, Doc.
Yes, sir.
Please, the assignment, are we going to prepare it in a slide form or in Word?
Very good. So please, strictly in a Word form. The font is 12.
12.
The font is 12.
Okay. Thank you, Doc.
All right. Okay. Right. All right. So please, let's continue. So functions of protein. We have the first column as the function of the protein. The second as the class of protein. Then the next one is example of protein. And then the fourth one will be the use of protein. So I'm going to mention the name and you read the very first one. First rule for me. So the function, you mentioned the function. The class, you mentioned its class. You mentioned the example, maybe only one example, and then one use. All right. So anyone to start with us? Anyone to start with us? The interesting thing is that the more you speak in my class, the more I know you. So, and it comes with a reward. So please, anyone to own up and speak?
Yes, Doc.
Yes, sir.
This David
Akum,
Akumunda.
Akumda, yes, I have seen you. So take the
The
first
first
one for
one
us.
is the function is metabolism. Into brackets the catabolism. So the class of protein is enzymes. Then the examples, we have hydrolytic enzymes. We have the proteases. We have the polymerases. We have the kinases. And then the uses. Cleaved polysaccharides, break down proteins, produce nucleic acids, phosphorylate sugar, protein synthesis.
Very good. Excellent. So in that line, you have hydrolytic enzymes, and the use of the hydrolytic enzyme is they cleave polysaccharides. When you come down to, you have proteases, and the proteases break down proteins. And then the polymerases, they produce nucleic acids. And then the kinases, what they do is they phosphorylate sugars, and they also synthesize proteins. Okay, very good. Thank you very much. Can anyone also pick the second one for us? Quick one.
Okay, defense. That's under function. We have defense. Then class of protein. That is immunoglobulins. Then examples, antibodies. Uses, we have mark foreign proteins for elimination.
Very
Then we
good.
also, okay,
Very good.
and
Anyone? Quick one. Another person.
That is cell recognition under functions. We have cell recognition. Class of protein, we have the toxins. Example, the snake venom, and they use block nerve function.
Very good. Very good. Can we have another person?
Okay, we have transport throughout body. That's the
Now I
function.
can see you. Now I
Yeah.
can see you.
Okay, so We have the function, we have transport throughout the body. Then class of proteins, we have the globins. Then the example, we have hemoglobin, the myoglobin. So the hemoglobin they use, we have carries oxygen and carbon dioxide in blood. Then myoglobin, the function is it carries oxygen and carbon dioxide in muscles. Thank you.
Thank you so much. Let's go to the membrane transports. Yes, who is helping us through? Roberts.
Thank you, sir. Function, we have memory transport. Then class of protein, we have transporters. Now when we come to the examples, we have sodium potassium pump, proton pump, anion channels. Then when we come to the uses, we have excitable memories, chemiamosics, and then electron transports. Thank you, sir.
Very good. But you medded the words like something. You are a chief medder. You are a chief medder. You medded the
It's
words like something.
chemiosmosis.
Chemiosmosis.
Yes,
And
sir.
then
Chief Meda.
anion, not anion, anion.
Yes. So the thing is with the examples, you said anion. It's supposed to be anion channels.
Anion.
Yes, anion channels. And then we have chemiosmosis. That's fine. But you did so well. Thank you very much. So we can move on. So it goes on and on. We
What
can't go
cancer?
through it one by one like that. So I think you can take time and go through yourself as well. I hope I'm clear. Do we need to go through it one by one like that again? We have more to
No,
cover.
please.
Right. So
That
I think
please is readable, so we will read.
read. So we can move on right now to other areas. So that should be the end of unit 1 for us with the second week. So we can now go ahead and look at unit 2. Unit 2. Right. So with the unit 2, we are now looking at genetic engineering tools. We will also look at agarose gel electrophoresis of DNA. We will look at recombinant DNA technology. And then we'll go on and look at methods for plant transformation. We will also look at plant tissue culture. And then finally, with the unit 2, we'll look at animal culture or animal tissue culture. So let's take the genetic engineering tools.
Hello, please, sir.
Come again.
Hello, sir.
Yes,
Good
madam.
evening, sir.
Good evening.
Please, the slide, will you share it or you shared it already?
Yes, just that I think unit 1, I have shared the unit 1 already.
Okay.
Up to the proteins. I mean, this side that you have on your screen, I have shared it already.
Okay. Thank you.
Right. So unit 2, let's now look at the genetic engineering tools. What are they? So genetic engineering, also known as genetic modification, involve directly manipulating an organism's genetic material. And in this sense, we are looking at DNA to achieve a desired result. So any attempt to modify a particular genetic makeup of an organism, you are actually embarking on genetic engineering. And that is what biotechnology is all about. We just engineer the DNA of organisms to produce some desired results or perpetuate desirable characteristics. Right. So this process allows scientists to alter the genetic material of an organism, whether plants, animal, or microorganisms, fungi, protists, prokaryotes, and the rest. Okay. In a precise and controlled manner that do not occur naturally. Now let's look at basic concepts of genetic engineering. First, what you have to do is that you have to identify the gene that you so desire. If the gene you are so desiring has to do with maybe being able to resist diseases, if that gene is able to resist droughts, if it's able to resist, salty environment, if it is able to resist pesticide, these are the genes that you are so much, interested in, and you want to insert it into another organism for the organism to exhibit that characteristics. So first, you have to identify that particular gene. The next thing is that you have to do gene isolation. Now, with the gene isolation, you use a particular enzyme called restriction enzyme to cut the DNA at specific sequences and allow for extraction of that particular target gene. And then the third one is you have to insert that particular gene into the desired organism or the target organism. And before you insert, we have so many methods of doing the insertion. You can directly insert and you can indirectly also insert. The direct we look at all the methods, whether direct method or indirect method. The indirect method, you can infect the organism with a bacteria, because that bacteria, you have inserted the gene in its plasmid, so that when you infect that organism, maybe plant or something, with that bacteria, the plasmid is going to be transferred into that organism that you want to transform, and it will pick up the DNA and it will start producing the proteins. That will produce the desired results. Okay, you can also have the direct ones where you use the gene gun. You can also use the electroporation method, and so forth and so on. These are all insertion methods we will look at in our later discussion. Then you look at gene expression as to whether what you inserted is being expressed. Now, the host organism that you have inserted the gene must show the characteristics that you have, inputted, or better so inserted in it. And then afterwards, selecting and screening is done. Not all the cells successfully incorporate the new gene. The cells that have taken up the gene are screened using marker genes, such as those conferring resistance to maybe antibiotic or something like that. Then you breed and test. Once the organism has been successfully engineered, it undergoes breeding, and testing is done to ensure that new gene works as intended and there is no side effects. The testing can include traits like growth rate of the organism. You can also, test for resistance to pests, resistance to, herbicides, resistance to droughts, and so forth and so on. Now let's look at some terms used in genetic engineering. Let's look at some terms that are used in genetic engineering. Now we have transformation, we have transgene, we have transformed, and then we have transgenic. We have many other terms as well that we will look at. So when we say transformation, we are referring to introduction of DNA into an organism. Transgene, we are referring to DNA introduced into cells of an organism. So the DNA that you are going to insert into that organism, we call it a transgene. Then transformed, the cells that contain the introduced transgene, or the cells that contains the introduced DNA, they are referred to as the transgene. And then, Ajekum, I think I can see your hand. Okay.
Yes,
So
yes, doc. Yes, doc. Hello,
yes,
doc.
sir. Yes, sir.
Yes, thank you. Please, so based on the genetic engineering, process, so does it mean that the immunization process, does it mean that it is the genetic engineering process that is used to do the immunization drug?
The immunization, do you introduce gene into the organism?
I think that I heard that that one is a killed, organism that is being used to make the drug, and therefore, so when the drug is being inserted into the body, that the children become resistant to that bacteria or the virus.
Very good. Now, question that I'm asking is that when you are doing immunization, do you introduce the organism, the inactive form of the organism into the human, or you introduce a gene into the human?
That is what I'm asking because I don't know whether it is the gene is being used in the drug.
Doc,
Very
is the
good.
organ-
So somebody is talking.
I said, Doc, is the organism, this is the organism, not the gene, but the organism in immunization.
The inactive form.
Yes.
Hello.
Doc, I said
Hello,
in
doc.
immunization, it is the organism, the inactive organism, not the gene.
Hello. Hello.
Hello, doc.
Yes, sir. Can
Hello,
you hear
sir.
me? Yes. I think I went off. Please come again.
Doc, I said in immunization, it is the inactive organism that is always being introduced, not the gene.
Yes, yes, that you are right. So the inactive form of the organism, if it has to do with bacteria, you are introducing the inactive form of the bacteria. If it has to be do with virus, you are introducing the inactive form of that virus. Okay. So you are not actually introducing the live organism into the human. When you introduce the inactive form, the body produces antibody to attack the antigen that you have introduced or a protein that you have introduced. So that is how immunization okay. But this time we are talking about introducing a gene, a DNA into the organism to transform it. We don't introduce vaccines to transform the organism. Please, I hope you get it.
Yes, doc.
Great. Joseph, your hand is up.
No, that is the audience.
Okay, so that's fine. All right, so I think we can continue. So transgenic, when you say an organism is transgenic, what does it mean? Any organism that is generated using genetic engineering technique is considered to be a genetically modified organism. In other sense, we say it is called GMO, genetically modified organism, or a transgenic organism. We also have common tools for the genetic engineering or for the genetic engineer. One tool that we have is enzyme. We also have vectors. We have hosts. And then we have techniques. And some of the techniques, we have the gel electrophoresis. We have blotting. We can have northern blotting, southern blotting, and then western blotting, depending on the kind of blotting that you want to do. And then, or the experiment that you want to carry out. And then we also have the polymerase chain reaction, PCR. I think this word became very famous during the COVID-19 era. PCR, they are running PCR. That's the polymerase chain reaction. We are going to look at that. And then we also have the recombinant DNA technology. That's the rDNA technology. And then the fifth one is commercial production. So first, you look for the enzyme that is going to be used. You have the a vectors that you are going to insert. The vectors can be a plasmid, and the host, after that, you introduce the vector into the host, which is a bacteria, it can be a virus. And then also the technique that you are going to use to carry out the process. And I have mentioned some of them already. And then you undergo the commercial production of such organism. Let's look at the enzyme first, because we spoke about the enzyme. Number one, the enzyme. So let's look at the enzyme first. So the enzyme, with the enzyme, we have the lysine enzymes. We have so many types of enzymes. So we have the lysine enzymes. Now, when we say lysine in biology, we are referring to cutting. So cutting enzymes, it's like a Caesar. Okay, we don't mean the enzyme is a Caesar, but I mean literally we say lysine, to cut. So if enzyme is cutting, we refer the enzyme to as a Caesar or molecular Caesar. Okay, so DNA and RNA are obtained from cells and tissues through extraction and purification of samples using a combination of physical and chemical methods. That is, the cell can be lysed or can be cut using several enzymes depending on the organism. The enzyme include cellulase, and I'm sure when I mention cellulase, you know what the enzyme will be used for: to cut cellulose. Pectinase for pectin, proteases for proteins, lipase for lipids, chitinase for chitin, lysozyme for other molecules. It can be any other bacteria or something like that. Okay, so we use those ones to, cut molecules of organisms, etc., etc. Now, we have endonucleases. They are also enzymes, and the endonucleases are referred to as molecular Caesars or restriction enzymes. Now, endonucleases are type of enzymes that cut DNA or RNA molecules at specific internal sites rather than at the ends of the molecules. So internally, when you look at the sequence that we have looked at in our previous lessons, gene, C, A, U, C, A, gene, like that, they are the sequences. So the endonuclease, endo means internal, so it doesn't cut the ends. It doesn't cut the 5 prime, towards the 5 prime end or the 3 prime ends. The endonucleases cut internally. They cleave the phosphodiester bonds with DNA or RNA molecules. An example of these molecules are the ECO R1, ECO R2, Hindi 3, SAL 1, BAM or BAM, capital H, I, and then we have the capital P, V, U, and then 2, etc., etc. All these enzymes were named by their discoverers. Okay, all these enzymes that we see, the endonucleases, are named according to their discoverers, or those who discovered them. So you can also identify or maybe discover your own enzyme and name it accordingly. Right. So the first isolation of DNA was done in 1869 by Friedrich Meister. Restriction is the process of cutting foreign DNA into small pieces. Most of them only recognize small short sequences. That is from 4 to 8 nucleotides. That's the NT. And they are referred to as the recognition sites. Recognition sequences are symmetric in that the same sequences of nucleotides is found on both strands. Both run in opposite direction. And if they are running in opposite direction, which means that if you have AAU, you are going to have UUA. So that is what it means. They cut in opposite directions. Restriction enzymes usually cut phosphodiester bonds of both strands in staggered manner. We are going to look at it. We have those ones that cut, in a staggered manner, others also cut, from top to down, and they all have specific names, so that the resultant double-stranded DNA fragments have single-stranded ends. So this is what we have. Now on your screen, we have a plasmid. Now with this plasmid, we have specific endonucleases for specific sites. So you have, well, if you can see on your screen, we have this endonuclease, we have this also endonuclease. I'm just circling them using my cursor. And then we have this one, 2, BG, II. We have this, we have cloning sites, we have the fusion site, we have this enzyme, all those nucleases that cut this specific site. They are site specific, so they cut specific portion of the plasmid. And plasmid is a circular DNA which is found in prokaryotes. Specifically, you are talking about bacteria. So these endonucleases cut specific sites. If you are using them, they cut specific sites. Right. So we have types of ends that are generated by these endonucleases. We have these type of sites that are generated by these endonucleases. Like I said, is somebody talking, please? Please, can I go on?
Yes, please.
Yes,
Yes,
please.
Yes,
please.
Doc.
Okay. So we have, please, I hope I'm not moving too fast. Am I moving too fast? Is it okay?
Yeah, it's okay.
All right.
But
Yes, sir.
I was asking whether the plasmid is the same as the nucleate in the bacteria.
No, plasmid is not the same as the nucleoid. The nucleoid contains the genetic material of the bacteria, but the plasmid is the circular DNA, which is found in the bacteria. So the area, the portion where you have the genetic material in humans or in other, higher organisms, we can see that it is the nucleus. Inside the nucleus, we have the genetic material. But in the, bacteria, you can't say it is nucleus because it is not membrane bound. So we can't refer to it as nucleus. So what we have to say is nucleoid. And this nucleoid contains the genetic materials. Please, I hope you are clear.
All right. So the DNA in the bacteria is the plasmid.
Yes, we have it as plasmid. And we also have some genetic material, which is DNA, in the nucleoid.
Okay.
Can I continue, please?
Yes, please.
Great. So we have types of ends that are generated by the endonucleases. And we have said that endonucleases are enzymes that cut internal portions of DNA. And so when they are cutting, you realize that please, you can mute yourself. So when they are cutting, they generate some types of ends. So this is a staggered end. You realize that the echo RR1, okay, the echo R1 is the restriction enzyme or the endonuclease which was used. So you have it cutting through between G and A, that is GA sequence, and then it comes and cuts this side too. So you realize that we have had a staggered form of cut for this particular, DNA strand. And we have also another type of cutting. It can cut from top to down directly. So you realize that the green portion on your screen, we have the enzyme cutting right from top to down. And then you have this portion, or the lower parts being separated from the ends. It's not in staggered form or conformation. So let's look at the use of restriction enzymes.
What I say?
So
Hello.
Yes.
The types of cutting doesn't have a name or there's a day. It's written there that do
So we
cutting.
have those ones that are, I mean, they cut from top to down, just like I showed you. This one. I don't know whether you have it on your screen. Yes. So you have it as, I mean, the top to down cuts, and then we have this one as the staggered cut, because you don't have it from top to down. We have some ends protruding. You have the yellow ends, I mean, protruding on equal fashion. But if you look at the second one, you realize that it is a from top to down cuts. Okay. So you see from the green portion that has been labeled, the cut was between the C gene, that's the cytosine, and guanine down to guanine cytosine. So realize both ends are equal, but in the other cut, you don't have it to be equal. It's a staggered form of cut, and the ends are protruding. Okay. So appropriate amount of enzyme is added to the target DNA in the buffer solution, sometimes supplemented with bovine serum albumin, which is referred to as the BSA. And the reaction is then incubated at 37 degrees Celsius over a range of temperature above to about 3 hours. And some reaction then have to be, I mean, heated and activated at 60 degrees or 65 degrees Celsius for about 20 minutes. That is when you are performing the restriction. Okay. Now, we also have another enzyme, and another name of this enzyme is the alkaline phosphatase. We've looked at the very first one, which is, I mean, the isonucleases that cause the ends. We have looked at the endonucleases that cause in between, and we have the alkaline phosphatase. They are also enzymes that is used as a tool in biotechnology. And what is this enzyme about? This enzyme is responsible for removing the phosphate group on the 5' of the plasmid to prevent secular formation. So like I showed you, the plasmid is secular in nature. And so this secular nature must be opened up. And the enzyme that you use to open up is the alkaline phosphatase. And it cleaves the 5' of the plasmid. Then the fourth one is the DNA ligase, which is responsible for repair and joining of two strands of DNA together by forming a phosphodiester bond between the phosphate group of one strand and the hydroxyl group of another. This essentially glues the DNA strands. So as the DNA has been cut, it has to, reform after you have added another plasmid or another gene of interest. So if you cut, you add the gene of interest, and then you have to join them, and you use the DNA ligase to do that. And then the fifth one is the nucleotide phosphorylase. This enzyme catalyzes the addition of phosphate group from inorganic phosphates, that's the PI, that's the inorganic phosphate, to a substrate. Now that we are done with the enzymes that we can use, for example, the alkaline phosphatase, the endonucleases, and the rest, we also have to look at for the vector that we are going to, after cutting the DNA, where are we taking the DNA to? We have to insert it in a vector, and the vector will serve as an intermediary where we can, that vector must be able to replicate the sequence, or better still, the DNA that you have kept in, and produce copies of it. And then you can use these copies to insert into a desired organism. And the appropriate one is, I would say plasmid. So cloning vector is the DNA molecule that is capable of replicating. And when we say replicating, we are talking about making copies. When we talk about replication, we are talking about making copies of that particular gene sequence that you have placed in it. It should be able to do that. If it is not able to replicate or making copies of such organism, then it is not a very good vector. All right, so it acts as a carrier molecule for the construction of the recombinant DNA. And ideally, it should be a small molecule to facilitate isolation and handling. And must have at least one unique restriction endonuclease recognition site. So we remember that, or better still, you remember that we look at the plasmid. And the plasmid, we had a number of recognition sites where a particular endonuclease can bind and cut. So what it's saying here is that that particular vector that you use must have a specific endonuclease that you can use to cut that portion, to insert your desired gene. So that is what actually is talking about. So there must be an origin of replication so that the DNA can be copied. So even as you insert your gene of interest into that particular vector, there must be an origin of replication that you can copy your desired gene. Then the next one is that it should have some sort of selectable marker that will enable it to be selected. For example, you should have maybe antibiotic resistance. So that particular portion of the vector that you have selected, or the vector that you have selected, should be able to resist a particular selectable marker. So for example, if you use ampicillin in that medium that you have placed the vector should be able to resist ampicillin. It should not die, or it should not malfunction. Another one is antibiotic. The ampicillin is antibiotic. You can have many other selectable marker. You can develop different selectable marker in order to be able to select that, okay, this is the gene that has been picked by this particular vector. Other than that, you will not be able to isolate it when you culture it in the medium, because the selectable marker would be able to isolate that particular, vector for you. All other vectors will not be able to function if you don't have a selectable marker for it. But that particular one that you have, which is able to resist that selectable marker, will be figured out, and then you can do your isolation at the lab. Common cloning vector are plasmid and phages. I have mentioned that already. So plasmid, you can use it as a vector, the circular DNA. And then phages. The phages we are referring to as, another name for phages is bacteriophage. So viruses that attacks bacteria, they are called phages. And then they can also have another name called bacteriophage. Now let's come to the next one that we can consider, the genetic tools for the engineer. Another one is the host cell or the host organism. So the host cell is a living system or organism in which the vector can be propagated. So after having that particular vector, you have to, and inserting your gene in that vector, that is a plasmid or the phage, bacteriophage, you have to insert that particular vector into a living organism or into a living cell. And it must be easy to handle and propagate it. It should also accept a range of vectors, the host cells that you are choosing. You should be able to accept a range of vectors. The bacterium Escherichia coli fulfills these requirements, and it is used in many cloning experiments. So E. coli that you have been hearing, E. coli, it's a gut vector. It's a gut. Your alimentary canal, it exists in your alimentary canal, which helps in digestion of leftover foods. So it is not really a harmful bacteria like that. So you can easily, and it is easy to manipulate. Okay, it's a bacteria. It's easy to manipulate, and they are not really harmful. So you can handle them in the labs, and they have very good. They pick up range of vectors. They are able to pick up range of vectors. So they become very, very, good when you select them for your lab work as a genetic engineer. Okay, so let's look at the techniques that are involved. Please, as we go along, if there is something that you don't understand, kindly prompt me and then I'll go back and revisit and then also throw more light on it. Yes, boss.
I think the
Yes,
bacteria
boss.
about bacteria are they not tiny microscopic organisms? So how do you lay hand on them with this work?
Right. Yeah, this is who we are. Why? Right. So what we are going to do is that we use microscope to observe them, powerful microscopes to observe them, and we culture them in a medium. So we are able to identify them, and we use powerful microscope to observe them, to view their internal structures, and also to break. We use some enzymes to break, if it has to do with, their outer covering. The outer covering, they have peptidoglycan covering, and so we'd have to digest the peptidoglycan, which is a polysaccharide, and then we can also digest all the proteins that surrounds it with proteases, and then we can now get hold of their plasmid internally, and then we can use endonuclease to cut the plasmid and insert our gene of interest into that particular vector, which is the plasmid, and then we can now pick up the vector and insert it into another gene, whether by using different techniques as well. So we use microscope. If you ask me, you cannot see. We use microscope. They are microscopic, so we use microscope to observe them. So let's look at the techniques. The techniques that are involved. So the techniques that are involved, one, I told you that already. Polymerase chain reaction. This term became a household term during the COVID era. So this is technique for amplifying specific fragments of DNA templates so that enough material or copies become available for subsequent use. So PCR, when we are running PCR, simply we are making more copies of the DNA that we have. When we are embarking on PCR, we are running PCR, we are making more copies of that particular gene. If it has to do with maybe antibiotic resistant gene, a particular anti-resistant gene, we will run copies of this, and that process is, we use PCR in running that instrument, PCR, for running that. Right. PCR was discovered by Caribe Mullis in 1983. Requirements for the process include source of DNA. You have to get your primers, that's the oligonucleotides. We have to get a thermostable DNA polymerase, that is tag polymerase, and a thermocycler. So it's a process that we follow, and then we make copies of this desired gene that we have. So this is the PCR machine. I'm sure you have it on your screen. Yes. This is the PCR machine that we have. This look more modern. This is the PCR machine that we use at the lab. So we can go on and look at the steps in using the PCR machine, or the PCR steps in running the PCR. So three main steps are involved. First step is we denature the DNA. Number two, we anneal it. And number three, the third one, most of the times, we don't really capture because you extend it. So we are going to concentrate on the two the denaturing and the annealing, and then I'll make comments on the extension as well. It's a very important process, the extension. So I'm still going to make mention of that. So DNA or denaturing is dissociation of DNA fragment into single strands by heating it. So we apply temperature to, break the hydrogen bonds that exist between the strands. I hope you remember the hydrogen bonds. The hydrogen bonds exist between the nitrogenous bases. So when you heat it up, what happens is that those bonds that exist between the nitrogenous bases will have to be broken, and they are the hydrogen bonds. And then when they are, broken, which means you're going to get you.
Doc, please, we can't hear you. Doc, please, we can't hear you.
Hello, can you hear me?
I went off.
Yeah,
Please, can you hear me now?
Yes, please.
we're clear.
Can you hear me now? Yes. Are you with me now?
Yes. Now you can hear you, sir.
Great. So we can continue.
The slide is off.
Please, did you hear the denaturing? Should I go back to denaturing?
The slide 2 is off. Ah, yeah, it has come.
Please, should I go back
Yes, sir.
to the
Yes,
Yes,
sir.
please. Yes, please.
Yes,
denaturing
sir.
process? Okay. So the annealing, what the annealing is done, please can mute yourself. The annealing is done at a temperature of about 60 degrees Celsius, and each strand base pair with a complementary primer flanking the region to be amplified. So what happens is that essentially when the denaturing, you increase the temperature to about say 90 degrees Celsius, 80 to 90 degrees Celsius, and then to denature it to form single strands of the DNA. And then when this hydrogen bonds between the molecules are broken, or the nitrogenous bases are broken, we say you have denatured it. And then you anneal, you allow annealing to take place. That's the binding of the nucleotides. Okay, the nucleotides will have to, bind, or the segment of the DNA that you have, the segment of the DNA that you have for copies of it to be made, will have to bind to the single strands that has been made during the denaturing process. And then when you are doing this, you have to lower the temperature to about 60 degrees Celsius so that annealing can take place, or binding can take place. Binding of your desired gene to the DNA strand can take place. And copy of it is made. If copy of it is made, then you extend the temperature, okay, so that the primer and then the polymerase copies of the rest of the fragment, in case some of the fragments were not copied, you extend it so that copies of it is made. Now, detection of the DNA product should be done using the fluorescently labeled dyes. You can use the SYBR
Hello, sir.
green. Yes.
Hello, sir,
Please, I'm listening.
please. What is the flaming area? I
Bernadette,
want to get it clean.
Bernadette, I'm listening.
Sir, please. I want to get it clean.
Bernadette, I'm listening.
Oh, I'm talking here. I didn't
Can
get
you
what you
position
were
yourself
saying.
well?
Yes,
Bernadette,
hello,
can
sir.
you
I
position
didn't
yourself
get what you
well?
were saying. Sir, I can hear you.
Okay.
Hello, please. Can you explain this? Explain a little further for the first one.
Okay. With
I
regards to
didn't
the
understand
annealing,
what you were saying.
is it the
Yes.
annealing or the denaturing?
Yeah, the first, the natural, I
Okay.
understand that the bonds would, but why would the nuclear, what do you call it? The after bonding again, why would the DNA polymerase come and bond to get another strand?
No, the DNA polymerase, you don't have the DNA. I mean, the DNA polymerase will not have to bind. The DNA polymerase, what happens is that they will, I mean, they would have to help in a sense, okay, in a sense, bringing the desired gene that you have. The desired gene that you have will be introduced into the medium so that the single strands will pick up this gene and make copies of them. I don't know what you understand. You have the DNA. The DNA, for example, the plasmid. The plasmid is a circular DNA, and you have your desired gene in a single strand form. The desired gene in a single strand form. Okay. Now, even if you have it in a double strand form, what happens is that during the denaturing, you have the plasmid which has been cut. In other sense, it is no more a circular DNA again. So it has been cut by endonuclease. So it is no more a circular DNA again. So that portion that has been cut, we refer to that site, recognition site. And as we started the lesson, we said that we have endonuclease that cuts specific sites. So that site has been cut. So we have, straight chain or straight double strand of DNA. No more plasmid, circular DNA. Okay, now you have your desired gene also in the medium. Now you increase the temperature at say 80 degrees, 90 degrees to denature both. If you have single strand of your desired gene, it will denature. The hydrogen bonds between the nitrogenous bases will have to be broken. Then you have two strands of your desired gene. Then the vector or beta, so the plasmid also will denature. The strand of the plasmid will also denature so that you get two strands. Now that you have four strands, if your desired gene was double strand, you have four strands. What happens is that you are now going to let your desired gene bind to the
Doc, please, you are off. We can't hear you. Please, you are off. We can't hear you.
Are you calling him?
Hello.
Please have patience. It's a network.
I'm trying to.
I dey go watch Champions League. Oh.
First, I beg, go call the man, make he can continue. Wait for after that,
The
then go
person
on
is
the
up
Champions
and you
League.
are still calling him. How can he hear
Eh,
it? You just wait for him
how
to make
can
the call and you come back, please.
he hear
Because
you?
of now his
The
phone
person
is
is
off. He
off.
can't hear
If
you.
if
We are
if
calling
you
him
go and
too.
watch, you go and watch.
Oh,
But he also come back and ask whether we are hearing him or not. So maybe upon saying it, you might hear and then know that there's a challenge.
Oh, he's not hearing you.
Hello? Hello?
Hi.
You say,
Yeah, please, those who are calling, you are rather interacting you are rather interrupting the network. Please stop calling. I've had calls and it has ended. I couldn't pick up, but you are rather interrupting the listening rather. Please, do you have the slide on the screen?
No, the
No,
slide
please.
is gone. The slide is gone.
Okay, please let me reconnect. Please, when it goes, cost rep, don't make an attempt calling because you rather worsen the situation because others will be calling and you also be calling and the this will break, the network will break. I don't pick calls when I'm presenting, so please don't call. Please. I'm trying to reset the network because it went off. So I think as we restore the network back, we can take a moment to, relax. We've done some 1 hour, 30 minutes already, so we can take time and relax and do the 1 hour left. And as we wait for the network to be restored, if you have any concerns, you can share with us. Yeah, I think one individual was asking clarification on the annealing and then the denaturing annealing and then the extension. I
Yes,
think I
sir.
can do that as we. Yeah, so with the annealing, I think it's the denaturing. I think that one is sorted, and please, I'm resetting the network. Just a minute, please.
Please, what is happening? I can't hear anything.
We are still waiting for Doc.
Okay, thank you very much.
We just have to wait. He says if I call, it's going to disturb his network, so I'm not calling. When he comes, we all join and we listen.
Okay.
When he said it, you all heard it when he said in class. So when he goes off, let us all keep waiting. When he comes, then we join the class. We are not going to call again because there when we call, it disturbs his network.
I nearly asked him what we should do in case he goes off again. Since
Let's
he
keep on
said he
waiting.
shouldn't
So
call,
if we
so what do we do?
cannot,
So you
it's
should
just a
rest,
matter of
so
waiting.
rest.
So wait, we heard him when he said we shouldn't call. He said the class rep is begging me that I shouldn't call again. So let's keep on waiting. When he joins, we all join.
That's all.
Yo, yate. Mr. Alima.
Adwuma ma ne nkyɛ kakra me kora metwe mu kɔ abɛdi mbanku mewie a na te sɛ mɛtwe awie no aba.
Ɛde asɛm koraa ni.
Nyɛ asɛm na abɛka. Ne ɛhoro hwee.
Hey, brother Victor near Bradriane. Oh no.
Manku ne dɔkono no saa deɛ wonya ama sa ɛno ano no ɛtitia. They keep you the whole day. Some like ɛtitia.
Victor, oh, Chiama,
Victor,
Yenina
can't you off your
Diana.
mic? Victor, can't you off your mic?
Hey, me mic is so. Ah, why not can't you off your mic? No? Okay.
You are disturbing us. Why?
Check. I don't know. Hey, I'm going to I did that.
All right. Hello.
Yes, dog.
Hi.
Yeah. Thank you very much. I would have to buy all of you yogurt. Seriously. One thing is that you see,
Dog, we are
yes. One thing is that please, apologies. I really apologize for the distraction in the network or the connection. One thing is that when I go off on you people keep calling, you rather worsen the situation. Others are calling me. I'm not home, so I'm using my, phone network to connect to my laptop. So when it goes off, when others call or when I receive call and it goes off and you also bombard it with more calls, you rather worsen the situation. So please, when it goes off, I will not pick that call, so it will not distract it. But when you fed that call, then it will worsen the situation by, letting it go off. We'll have to reset again, and it will quite, delay it a bit. So please take that note for me. Then,
Hello, sir.
on that said and done, kindly accept my
Hello,
apology.
sir.
Yes, yes, sir.
Apology well accepted and your point well noted. So how can we notify you that you are off if we say we shouldn't call?
Kindly send me a text, or better, you can WhatsApp me. My phone is on. My phone is on. So when you send me a message, I will receive it. Okay. When you notify me through maybe a text or WhatsApp, I will be able to read it.
Okay.
Great, great, great. Thank you so much. Thank you so much. Right. Please, I think we can now move on. Right. So yeah, back to the question that somebody asked after restoring the network. Back to the question that somebody asked with the annealing, the denaturing, annealing, and then the extension. Let me be quick on that, then we can continue. So denaturing is essentially breaking the hydrogen bonds that exist between the strands to make your single strands. And then the annealing has to do with you have your desired gene already. That is sequence of those nucleotides already in that medium. So you want to make copies, essentially. You want to make copies of that particular gene of the desired traits. So we want copies of that. So you allow it to bind, whether it is a single strand or double strand. When you denature it, that one to the bonds that exist between it will be broken to make it single strands. And if it is single strand already, what you have to do is that you anneal by lowering the temperature. Annealing simply means you want to, let your desired DNA bind to the plasmid that you have, okay, the copy of the plasmid that you have. So you lower the temperature so that it can bind. When it binds, copy of it will be made. If you have only a single strand and a copy is made, you are going to get two strands. And another cycle of the PCR will give you three. When you have two, you're going to get four. Another cycle of the PCR will give you 8. Another cycle will give you 16. Another cycle will give you 32. Like that, like that. So if you have 3 cycles, you are going to get 32. So if you have 4 cycles, you are going to get, I think, 64. When you have 4 cycles, or sorry, 5 cycles, you're supposed to get 100 and I think 28 or something like that. So that is how the cycles run. Okay. Yes. So the cycle is such that you denature, you anneal, and then you extend. When you extend, it means that you still continue to lower the temperature so that other ones that haven't bound it yet will continue to bound. And a copy of it is made. If a copy of it is made, you denature it again by breaking the bonds. When you break the bonds, then you allow annealing, that is binding, to take place again, and then copy of it is made again, and then you extend it, you denature it again to run another cycle. So the cycle is to just make copies of the DNA or the desired gene that you want to, I mean, to transform other organism with, so to speak. Right. So that is the PCR. So we want to detect the copies of that particular gene, the desired gene. How do we detect it? We use the SYDR green. That is an intercalating agent. So it's a dye. Essentially, it's a dye. So you have to, I mean, use the dye for you to be able to identify it in the medium. So as the PCR progresses and the quantity of the double-stranded DNA increases, more dye bind to the PCR products, and hence the signal intensify and also it increases. Since the dye bind to all amplified PCR products indiscriminately, what happens is that you have to, I mean, subject it to something called melting curve analysis. And it's carried out to detect the non-specific DNA product or the product that is being produced. So the melting curve is used to actually, I mean, detect what you have. So this is the polymerase chain reaction I have been talking about. So first, you have your desired gene in red. Okay, you have your desired gene, that's the primer. Use the primer to bind the first sections, or better, the first few, I mean, sequences. So you have your nucleotides in yellow. So these nucleotides will arrange. Now, if this nucleotide arrange, that's the second step. You have your primer that will bind, and the nucleotides will also be arranged. Now, if your nucleotides bind to it with the primer, it will be copied. Now, if you look at the last end of this diagram to the right, you realize that at the end of the day, how many copies have been made? Now you have the first one, the denaturing. Let's go to the left side again on your screen. You have the very first one, that is number 1. You have it 5' to 3'. And then down there, you have the complementary base pair, which is 3' to 5' in the opposite direction. 2 strands of the parent DNA. Now, with the second one, you have the primer binding to it. That is the primer that you are going to use to start the sequences, right? So with the third one, you have the arrangement of, or the binding of the nucleotides in yellow. The nucleotides are in yellow. They will bind to form sequence, and they will run from 5' to 3'. As always, that you know, I mean, the copying of the DNA from the 3' to 5', always, continuously. So we come down here. That one too, the nucleotides will bind. The nucleotides will also bind. That's the third point. So when you finish, what happens is that you are going to denature it again, which means that you're going to break the bonds again up until we get the last point. You break the bonds again between, I mean, the third point, okay, this has annealed, or better still, it has bonded to the parent strand. So up here, down, and then we come down here to look at it. So the primer and then the nucleotides have arranged nicely to form a strand, a new strand. When you go up here to the primer and the nucleotides have also arranged to form a new strand nicely. Then you go back again to the PCR cycle, denature it again. When you denature it again, the bond will be broken. You want more copies to be made. So you denature it again to release the strand that has, I mean, formed from the parent strand. And then you anneal it again. When you anneal, what happens is that you are going to get more of the nucleotide also binding to the strands that are found in the, I mean, the medium. Then you denature it again to break, after forming the new strands. You denature it again, break the bonds, and then you allow the temperature to come down to anneal, for the nucleotides to anneal. and then to form a new strand. When they form the new strand, then you denature it again. So upon how many cycles that you want, maybe 30 cycles, maybe 20 cycles, maybe 15 cycles that you want the machine to run, it will give you the number of copies that you so wish or you desire. So essentially, just as I said, we run PCR to make copies of the DNA that we want to insert into the organism that we have targeted. I'm sure it is clear. Is there any other clarification that you would want to seek?
Say,
Hello.
say,
Yes, Bernadette.
the genes that in the machine.
Come
Now,
again.
they need, I said, this,
Bernadette, your network is very bad though. It keeps breaking.
when you get to the intro, this thing is done in the machine. Now, what would we get the nucleotides to the parent apple, then to get that numerous thing?
So we have a sample. Okay, we have samples that we feed into the machine. We have samples that we feed into the machine. The nucleotides, they sell them.
That is where my confusion
The
was. Thank
primer, the primer and everything. The primer and everything is sold.
you.
Is that okay? We have industries that produce these things. The primer and the rest. Okay. Those
Okay, so thank
reagents,
you.
I mean, those reagents that you are going to need for this process, actually, they are sold. We have manufacturers that produce these things that you feed into the machine, and they will do the work for you.
Okay, thank you, sir.
Good. But what you need is your desired segment of the DNA. Your desired segment of the DNA. Is that okay? Your desired segment of the DNA that you want to insert into the organism, so that copies of this will be made for you in the PCR machine. You need that particular segment. Are you okay?
I'm okay, sir. I'm okay, sir.
Very good. Very good. So you extract segments of the DNA from the organism that you have desired the gene from, and then you isolate it. After isolating it, then you run PCR to make more copies of the DNA that you have extracted. But for the reagents, it's sold, so you can get them, and then you feed it into the thermocycler, which is the PCR machine, and then more copies of it will be made for you. Right. So let's go to the second one. I think we have kept a bit long here because of our network, but then we can still make headway. Gel electrophoresis is another, technique that we use. So PCR, you run PCR, you also do the gel electrophoresis. Now, we use the gel electrophoresis to segregate segments of the DNA. Now, DNA is negatively charged. Okay, DNA is negatively charged. So when you feed it into the electrophoresis machine, or better still, the chamber, what happens is that it's going to, and you plug it, you apply electricity or voltage. What happens is that it's going to travel from the negative node to the positive node. Then when they are traveling, the heavier one will not actually be able to travel faster, but the small segments of the DNA will be able to travel faster because of their molecular weight. We'll look at it briefly, how this all pans out. So according to physical properties such as the size and electrical charge, we apply the electrical current to them. And the current forces the molecule through pores in a thin layer of gel, a firm jelly-like substance. And that substance is called agarose gel. You can use other gels for it. The gel can be made so that its pores are just the right dimension for separating molecules within a specific range of sizes and shape. Smaller fragments usually travel faster than the large ones. I have said that already. So what is the purpose of the gel electrophoresis actually? To visualize nucleic acid, that is the first purpose. Also to quantify the molecules, whether, it's bigger, it's a smaller, and all that, to quantify it. And then also to isolate a particular band. That is where our interest, to isolate a particular band of the DNA. And also to score for the presence or absence of expected band, whether a band that we are expected or we are expecting is in there or it is absent or it's not there. And then also determine the size of the separated bands by comparing to the bands of known length or the size. The types of gel that we can use the agarose gel. We can also use the polyacrylamide gel. We can also use the starch gel. But that one depends on the kind of experiments that you are using. So the agarose gel, when do we use the agarose gel? The agarose gel are run horizontally when nucleic acid separation is based on size only. So samples are also easily recovered, and after the experiment, the resulting gel can be stored in a plastic bag in a refrigerator. Now, this base gel separation of DNA fragments ranging from, say, 50 base pairs to several mega bases, that's millions of bases. It is the easiest and the commonest way of separating and analyzing DNA. So we use the agarose gel most of the times in our lab, not the polyacrylamide and then the starch gel. Yes, they also have their side, their advantages, but then most preferred one is the agarose. The only advantage is that it takes long time for it to run. And another problem is that it is quite fragile when you are handling them. So the time of running it is quite long. It takes quite long for you to run it, and then also the handling is quite fragile. So if you don't handle it well, it becomes problematic. And then high percentage of gels are often brittle and do not set evenly. Then let's look at the second gel. That's the polyacrylamide gel. And this is used for separating protein ranging in sizes from 5 to 2000 kDa due to the uniform pores size provided in the gel. So before you run this, you have both the agarose and then the polyacrylamide. You create wells. But for this, due to its uniform pore and size provided by the gel, this is also much preferred. Now, pore size is controlled by controlling the concentration of the acrylamide and base acrylamide powder used in creating the gel. And care must be taken when creating, or care must be taken and must be used when creating this type of gel. And as acrylamide is a potent neurotoxin in its liquid and then powdered form, so it can influence your neurons. The operation of your neurons, transmission of nerve impulses can influence that. Starch gel. So partially hydrolyzed potato starch makes for another non-toxic medium for protein electrophoresis. and the gels are slightly more opaque than acrylamide or agarose, as we have discussed. And then non-denatured proteins can be separated in starch gel according to charge and also the size. And they are visualized using the naphtha black or the amido black staining. Typically, starch gel concentration are 5% to 10%. As the agarose gel, we use the green to visualize it. So materials required for the gel electrophoresis, you have the loading buffers, and the loading buffers gives color and density to the sample to make it easy to load into the wells. And then also the dyes are negatively charged in neutral buffers and thus move in the same direction as the DNA during the electrophoresis. This allows one to keep track of the position of the DNA in the gel. The most common dyes are the bromophenol blue and also the xylene cyanol. So if you are preparing the buffer, the buffer that we just spoke about, you would need this. I don't need to go through all of them, but then unless otherwise you are at the lab and I'm taking you through how we prepare the loading buffer in gel electrophoresis chamber. But then aside that, we can just mention few things here and then move on. I am not going to ask you how do you prepare for the loading buffer or the recipe for ask you for the recipe for loading buffer. But it's just an extra information that I needed to give you. That's why I have brought it in the slide. Is somebody's hand up? Right. So the recipe for loading buffer, I am not going to ask you, or how to prepare for the loading buffer, I am not going to ask you. But then, of course, you need to know something about the loading buffer because the requirements for the gel electrophoresis. Okay, determining the size or the charge or getting the sample length of your DNA and all that. Loading buffer is important. So 25 milligram bromophenol blue or xylene cyanol, 4 grams sucrose and in water to 10 ml. And examples are the trace acetate EDTA, that's the TAE, trace borate EDTA, and then sodium boride. You also have the TAE has the lowest buffering capacity, but also provides the best resolution. This means a lower voltage and a more time, but a better product will be formed with the loading buffer. So SB is relatively new and it is ineffective in resolving fragments larger than the 5 kilobases, but it has the highest buffering capacity, allowing voltage up to the 350 volts. So essentially, the gel percentages that you need are all provided in the slide in case you want to have to read for further understanding, or better still, for maybe you want to prepare a loading buffer and you want to go through, you can do justice to it. If you have time to, you can come around and we can do the preparation of the loading buffer for the gel electrophoresis. But the percentages here have been given in your slide. So the gel tank, the description of how the gel electrophoresis tank and all that have been given. Bashiru, your hand is up.
Yeah.
Bashiru.
Please, yes, yes.
Yes, please, Bashiru, I can hear you.
Please, the gels, can it be found in the market?
Yes,
All the gel
yes,
you mentioned.
we can buy them, but you have to prepare it yourself. When we have the composition, the recipe. Okay, we have the recipe
Yes.
I mean, if the starch gel, the polyacrylamide gel, and then the agarose gel, they are all in the market. They are manufactured by companies. So if you buy them, you have to prepare it yourself. Okay.
Okay.
So you have to
Yeah.
prepare it yourself at the lab before you load it into the electrophoresis chamber. You have to prepare it yourself. But the this thing is there.
Okay. Thank you.
The this thing is there. The reagent is there. It's loading them into the gel electrophoresis chamber. Then you can run your gel electrophoresis. So the tank that you are going to use, I think I have the tank here. So you can look at the tank. This is the gel electrophoresis tank or the chamber. That you prepare the gel, you pour it in there, and then you allow it to cool to solidify. So this is the tank that we use to prepare. We keep the gel in after preparing it. We keep the gel in, and then we can connect it to electrical currents. When it's in, when it's connected, the DNA will run from the negative end to the positive end because it is negatively charged. DNA is negatively charged. Consider the size, the description of the size of the gel tank that you see on your screen. Okay. We also have markers that we use to determine the sizes of the DNA which is running, or the molecules. If you are using RNA, the molecules that is running in the gel. So they are set of standards that are used to estimate the sizes of DNA fragments. These are the markers. And this give bands with known sizes. So we use the markers to determine the sizes of your DNA which you are running in the electrophoresis chamber. Okay. Now let's look at the equipment and reagents for running the gel electrophoresis. We have the electrophoresis chamber I have showed you already, power supply, and then gel casting tray. It's also in the chamber. Then you have the sample combs that you are going to use to create wells that you are going to use. The wells is where you are going to load your DNA samples in the agarose or the polyacrylamide gel or the starch gel. So it continues to tell how you are going to carry out the gel electrophoresis process. And this is more practical than the theoretical way of describing it. But then I still want to go through with you. So gel electrophoresis buffer, usually trace acetate or the trace borate, is what we're going to use. And loading buffer with glycerol or 1 or 2 tracking dyes are also used. And then the fluorescent dye to stain the nucleic acid. Example is the ethidium bromide. And I have to tell you that the ethidium bromide is carcinogenic. So when you are using it, you have to be very careful. In case you chance on one and you are using, you need to be careful. And then you can also use the SYBR green to also stain the nucleic acid or the DNA that you are running the electrophoresis on. Now, we also use the trans illuminator to visualize the sizes of our DNA. So this is it. I have brought the setup again. So you realize that if you look at where my arrow is rolling on your screen, you have the wells. The wells are where you see black dots on your screen. Vertically on your screen. It runs from top to down. So these are the wells that you create with a comb. It's not a comb that you use to comb your hair, but then we have a special comb that we use when we are preparing or after preparing the gel and we pour it into the electrophoresis chamber. And this is the electrophoresis chamber. This is the electrophoresis chamber. And you have your agarose or the acrylamide gel or the starch gel in there. So you create the wells in there before they dry up or solidify. And so when they solidify, you have the wells in there. And then you put, or you, what you have to do is you introduce your DNA samples in the wells. And after introducing your DNA samples in the well, then you have your buffer. You pour your buffer. I have described the buffer in a previous slide. So you pour your buffer, or the recipe for the buffer too, I gave it to you. You can go back and read. So you pour your buffer, make sure that it submerges, the buffer submerges the gel containing your DNA samples, and then you connect it to a voltage, or better so to a current, and then you plug it on. When you plug it on, you realize that there will be, movements. There will be movement of these samples in the wells from the negative, side to the positive side. But the traveling will be done according to the sizes of the DNA that you have in the wells. So if it is larger, it may not be it may not be able to travel faster as compared to those with, lower molecular weight. So the steps involved are what has been described in the slide. So you can go through. So I think I have basically described the steps already using the machine, the electrophoresis chamber. So kindly go through and read. So let's look at the recombinant DNA technology. If we want to look at the recombinant DNA technology, the stages that are involved. One, we have to cleave the DNA, that is, we cut. If you want to introduce the DNA into a sample, that's the technology. We want to introduce the DNA into a target organism. That is the technology, DNA recombinant technology. So the first stage is that we have to get our DNA cleaved or cut. And then number two, we produce or production of the recombinant DNA, and then we clone it. After cloning, we screen it, and then finally we insert the gene into the organism, or working with the clone gene, no matter how you prefer it to work with. Whether you introduce it into the organism after screening and cloning, or you use the clone to perform other experiments. Right, so let's take it one by one and look at how this pans out with the process. So a restriction endonuclease, we have mentioned the endonuclease, the function of the endonuclease in our previous slide already. So a restriction endonuclease is used to cleave the source DNA into fragments, into pieces. Okay. The restriction site likely to occur many times within the source DNA, so that cleavage will produce large number of different fragments. And I have also told you the restriction site. We have enzymes for specific restriction sites that they bind and cut. So when they bind and cut, you may have possibly many of these fragments or different fragments forming. Now, after this, now that we have just described electrophoresis, using electrophoresis chamber or the machine, the fragments can be separated from one another according to their sizes. I have told you that we use the electrophoresis chamber. You have your gel prepared as to whether you are using the acrylamide gel, you are using the agarose gel, or you are using the starch gel. What happens is that you prepare it in a liquid form, then you pour it into the chamber. I'm going through the steps again with you. because I didn't describe it using the slide, but I used the chamber to describe it for you. The description are on the previous slides, so you can take your time, go through, and then read. So after preparing it in the liquid form, what happens is that you pour it in the chamber, and after pouring it in the chamber, you make sure that you have your comb in there to make the wells, and then this fragment that you have clipped, you feed it into these wells, and then you pour your buffer on it and cover it and connect it into a voltage, and they will start traveling. After owning the voltage, they will travel according to their size. The speed of travel will depend on the size of the molecule that has been cleaved, or the species of fragment that has been cleaved. Right. So production of the recombinant DNA. The DNA fragments are inserted into plasmids or viral DNA, which have the same restriction endonuclease as the source DNA. Now, when we say source DNA, we are referring to the DNA that is having our desired gene, the gene that we so desire to have. The organism, producing characteristics of it. Now, we have to screen it. After getting the fragment, we need to screen it. The clones that contain specific DNA fragments of interest, and for that matter, about a particular gene, are in a mix of others within the library. If you have a lot of, what this means is that if you have a lot of fragments of DNA, but you don't have, or you are unable to identify a particular one that is of interest to you, we call that one a gene library. A gene library. So you have to screen that to identify the one that you, or that gene that carries, your particular trait of interest. So they are identified through screening. That's the next point. And how do we do the screening? Screening can be achieved by making the desired clones resistant to a certain antibiotic and giving them other properties that makes them readily identifiable. So I think I have spoken about this already in our previous slide, that when you insert it into a plasmid, you subject that plasmid, okay, a particular gene that you have inserted in a plasmid, you subject that plasmid to a medium of a particular antibiotic, or it can be any other selectable marker that you want to use. Then when the desired gene, or better so the desired plasmid, the plasmid that contains the desired gene is able to resist that antibiotic or that selectable marker, then that is possibly your gene of interest, and you can comfortably or readily identify and get it out of the library that you have. So we can have modes of screening, different modes of screening. And you can use antibiotics, like we have said. The antibiotics, we have ampicillin, we can have different antibiotics. Depends on the particular bacteria that you are working with. So first, preliminary screening of clones is done to eliminate from the library any clones that do not contain vectors, as well as clones whose vector do not contain fragments of the source DNA. Only those clones that contain the vector will be resistant to the antibiotic and will be able to grow on exposure to it. So if it is not, resistant to it, it will surely die off, or it will cease growing. But if it is resistant to it, which means that is your desired gene, it will be able to grow even after you exposing it that particular antibiotic. You can also use the colony hybridization, where a clone library could contain several thousands of fragments of source DNA, and parts of the nucleotide sequence of the DNA of interest is known for a probe construction. And the nucleotide sequence of the probe is complementary to the gene of interest. Now, what happens is that the probe is expected to hybridize with the complementary single-stranded sequences on the bacterial DNA. And then colonies that contains the gene of interest hybridized with the radioactive probe and emit radioactivity on the film. So if it is not having, that colony is not having the desired gene of interest, it will not be able to emit any radioactivity on that film that you are using. Now, let's look at the methods of the transformation. And specifically, we are using that of plant transformation. How do you do that? Now that we have gotten our gene of interest, how do we then feed that gene of interest into the organism? And specifically, we are talking about plants. How do we transform the plants? I think we have defined the terms already in our previous slide. Transgene, transformation, transgenic, and the rest. We have defined them already in our previous slides. So, methods of plant transformation. How do we change the genetic composition of plants? That is the meaning. So, transformation is biotechnology, and it's a process of introducing DNA into plants and animals. There are two main methods that we can use. We can use the indirect one. Another name is that we can use the agrobacterium-mediated gene transfer, where we infect the plant with a bacteria. The agrobacterium is a bacteria, and the full name is called Agrobacterium tumefaciens. And we can use that as a mediated gene transfer. And then we can use the second one, which is the direct introduction of the DNA into plants, where we can use the gene gun. We can also use the electroporation. We can use many other methods that we are going to look at in our subsequent slide. Now, the agrobacterium-mediated gene transfer. So, Agrobacterium tumefaciens, as I said, now it has an updated name. The name is the Rhizobium radiobacter. Rhizobium radiobacter. And it's a plant pathogen with the following characteristics. 1, it is gram-positive soil bacterium. 2, it causes disease called the crown gall. And then 3, diseased plant develop tumors at the crown. That is at the soil level, just above the soil level. Now, disease which is caused by this bacteria is by stable integration of the bacterial DNA into the plant genome. And natural occurrence of the disease is restricted to dicotyledonous plants, which means that it will be quite difficult for you to use or conduct this experiment using monocots. It really works well with rather the dicots, not with the monocots. Right. So essentially, this is what we have. I mean, I'm going to summarize the whole thing with this diagram that you have on your screen. So first, you have the bacteria. You have the bacteria, which is the Agrobacterium tumefaciens, and you have the plasmid. If you look at it carefully, you have your plasmid in there, and you have your nucleoid. One gentleman acts of the nucleoid. So this is the nucleoid of the bacteria. if you look at where I am scrolling my cursor, the nucleoid of the bacteria, and aside the nucleoid, we also have our circular DNA, which is the plasmid. Now, what we have to do is that we break the bacteria apart using, several enzymes. Okay, different enzymes, because this wall contains protein, it contains polysaccharides and the rest, and each of them needs a specific enzyme to break it apart. So we insert a desired gene, okay, after lysing the plasmid. What we will do is that we insert a desired gene in the plasmid. If you look at it, a restriction enzyme was used on it, and the gene has been cut. The plasmid has been cut, exposed. It's no more circular again. So this portion which is exposed, we can now fit our fragment of interest, the DNA fragment of interest, into the DNA, the plasmid. So realize that at the end of the day, you're going to get a plasmid which contains the gene of interest that we have. Now, the gene of interest is colored pink, as seen on your screen. Okay, now we have had stable integration. We have had stable integration of such gene into our plasmid. Then we introduce the plasmid again into a bacteria. So down here, if you come down here, please, my cursor is rolling down here. So if you introduce the plasmid back into the bacteria, you know the generation time of bacteria, the growth rates, reproductive rates of bacteria is about 20 minutes. Okay, in every 20 minutes, they reproduce. So it will be able to reproduce, and more copies of this will be made. Now, you can now infect this particular plant cell. Okay, you can now infect this plant cell with the Agrobacterium tumefaciens. You can now infect this particular of interest, and it will be picked up by the plant, and the plant will grow, and at the end of the day, start producing the characteristics that we so desire the plant to have. So it is very simple. Let's look at the technology. First, you have the bacteria, which is having the plasmid, and the plasmid is serving as our vector. So we take the plasmid off from the bacteria, and then we use restriction enzyme, which we have discussed already, to break the plasmid apart. We cut it open at a restriction site, and then we insert our gene of interest there in the restriction site. And then again down here, we insert the plasmid back into a bacteria, and then infect the plant with that bacteria, so that at the end of the day, the gene of interest will be in the plant, which will be grown, and it will start producing those characteristics that we so desire for the plant to have. Please, up to this point, any question? Is it any question? Please, are you with me?
Yes, Doc.
Yes, please, we are with
Okay. Right. So we will end here, but then let's look at the agrobacterium, how you transfer it. I have described the process already, but let's just look at how this whole process works, look at the characteristics, and then we can look at the direct method later when we meet Saturday, God willing, and go on and finish up with the animal tissue culture, and then we end unit 2, and probably we can start unit 3 on Saturday as well and finish up the unit 3 isn't, I think, more distant as compared to the Unit 1 and Unit 2. So I'm hopeful that by Saturday we can meet, and if we don't finish up with the Unit 3, it will be left with something few, and then we can meet and finish up with it before we start with your quizzes. Right. So let's have the steps. I have described the steps already, but let's go through the steps again. So first, if you want to use the bacteria, transform your plant through the use of the bacteria, Agrobacterium tumefaciens, what you have to do is that first, you clone the transgene. I am not going to explain the transgene because I have defined them, all the terms, in my introductory slide. So you clone the transgene into a suitable vector. And a suitable vector we are referring to as the plasmid. Now introduce into the Agrobacterium strain. Number 3, you incubate the bacteria with a plant tissue. So after incubating it with a plant tissue, you wash and you incubate in a culture medium. So the last batch one. So the last batch one, which is the fifth point, I'm rolling my on it, my cursor on it. This is the medium. Then you transfer to regeneration medium and a high oxygen for root and high cytokinin for shoot initiation. So these are plant hormones. You introduce the oxygen for root and shoot elongation, and then also the cytokinin for for cell division. This is for cell division of cells. Okay, for shoot initiation. It helps in the meristematic regions, the cytokinin. Now let's look at the advantages of agrobacterium mediated methods. Now, the agrobacterium mediated method, 1, it has a wide range. It has a wide range of hosts. So it can infect a number of plants as well. Now, B, it has large amount of DNA that can be introduced. Okay? It has large amount of DNA that can be introduced. Better so, you can introduce the desired gene, no matter how large it is. This particular bacteria will be able to take it up. And then number 3, it is safe and easy to set it up. The method. And D, no costly equipment will be required. The equipment that is going to be used, they are not that costly. And the efficiency of transformation is high. It is able to take up those genetic material that is of interest to you, unlike other species of bacteria. And there is stable integration. That is, it has no complex and multiple integration. The integration is very stable, and the plant is able to produce those characteristics that you so desire. I think in everything, there are some disadvantages that are associated with it. So let's look at some disadvantages that are associated with the such medium of transfer. That is the use of agrobacterium. One is that it has limited application, and that is integration into plasmid not possible. Integration into plasmid may not be possible. So you realize that it's not all the, gene of interest that will be taken up by plasmid. So this is a limitation. If it is the plasmid is able to take it up, then there's high possibility that we are able. to integrate it into the bacteria and infect the plant with it. But we may have challenge integrating the desired gene into a plasmid. Number two, targeting into a selected tissue not feasible. So realize that plants we have number of tissues that makes up the plant. Infecting the plant with the bacteria. The bacteria may not infect a targeted tissue. For example, if you want to target the parenchyma tissue, the bacteria may infect the collenchyma. It may also maybe infect the sclerenchyma instead. It may also infect the phloem tissue or the xylem tissue or any other tissue, the meristematic tissue, instead of the targeted tissue that you want to infect. The bacteria may not infect the targeted tissue. So that is another challenge that we resort with it. So on that note, we would want to end the class here with the indirect method, and then we can continue Saturday on the direct mode of introduction of DNA into plants. Thank you very much for your time and your patience, even when the network went out. I'm so much grateful.
Thank
Is
you.
there a question?
Thank you too.
Bashiru, your hand is up.
Yes, in the
Bashiru, your hand is
lectures,
up. Yes, sir.
yeah, yes, yes. Yes. You mentioned that common cloning vectors are plasmids and phages. And you said the phages
Great.
has another name. Which, yes, that name that I wanted to, another name for the phages.
Bacteriophage.
Bacteriophage. Okay, okay,
Yes.
thank you. Please, under the tool for genetic engineering tool, we mentioned 5. Is it that we have more, or is it the 5?
Oh, these are tools that you are going to, require. Aside other sterilizing agents and those kind of things that you are going to, essentially, these are tools that you need. The enzymes, the vectors, and then those techniques that you are going to use as to whether you are going to use the gel electrophoresis, the PCR, and those kind of things. Okay? These are essential tools that you need. These are essential tools that you need, aside other equipment that you maybe sterilizing agents and those kind of things. You may even use the flow hood. Okay, when you come to the lab, you may use the flow hood, you may use the gloves, you may use and those kind of piston and mortar and those kind of things to grind your plant material for extraction of the DNA and all that. Those ones we don't really pay attention to. But the major tools that we use or we are interested in, are the vectors, are the enzymes, are the techniques that you are going to use, and the other rest that I have mentioned on the slide. Please, are you okay with that? Great. Right. I guess there is no other question. Is it in unless otherwise there is any? If there is no other question, I think Cosrep, I'll forward the slides to you. Unit 2, the entire slide. I'll forward it to you. I'll forward it to you. The entire unit 2, I'm going to forward it to you. I think we have done we have done the plant tissue culture. So we are almost done. We are almost done with the plant tissue culture. Then we go to the animal tissue culture, and then we'll end with that one for the unit 2. And then we can move on to unit 3 during our Saturday lecture. So thank you very much for your participation. And then I'll forward the slides to you just as I have promised to your course shop, and he will share it with you. God bless you and have a very wonderful night. Please don't hesitate if you have any challenge with any of the slides or with any other topic or session that I have gone through with you. Let's share. I mean, you can bring your questions on. You can type your questions and send it to me personally or even on the page, and then your course rep could get answers from me, and then we'll share with you as well. If you want to contact me personally, to no problem, you can feel free and
Hello, sir.
contact me for clarification.
Hello, sir.
Yes, sir, boss.
With the assignment that you gave that we are submitting on Saturday, are we supposed to submit it individually to your email or?
Yes, I provided my email, so you're supposed to send it
Okay.
through the email.
Directly to you.
Yeah, but it should be, please don't type directly in the mail and send.
No, so as a Word
Please
document,
don't
and
type
mail it to you.
directly. Yeah, it should be Word document with your index number.
Okay.
Word document with your index number and your name.
Font
Word
12.
document with your
Font
index
size
number and
12.
your name. The font size is 12. The font is Time New Romance.
Time New
Time
Roman.
New
All right.
Romance. Yes.
I needed you to repeat because some joined later. That is why I'm asking all these questions.
That's okay. That's okay. No
All
problem
right.
at all.
Thank you, sir.
You're welcome, sir.
Bye.
You're welcome. Okay. All right. Have a good night.
Same to you
Sounds
too. Thank
like
you, Doc.
Thank you. Bye. You're welcome. You're welcome, sir. Bye.
Mr. Ajimai, please, what is the assignment? Some of us couldn't join that day.